Hello, everyone. I'm Mike Moore. I'm a professor of biology at Oberlin College, and I'm very excited to be here today to talk to you about some of the challenges we face using genomic data for understanding relationships among closely related species. So this picture you see in front of you is a very special place. It's the greenhouse of Ann Sakai and Steve Weller at the University of California, Irvine. And all of these plants you see here are different species of Sheedia, which is the project that we were working on together. And I was very, very lucky to be able to visit this greenhouse. And you can see a fairly large proportion of the diversity of Sheedia here. And so a question that I personally am interested in my own work is how do we actually know the evolutionary history of a group like Sheedia? And more specifically, how do we understand the relationships among closely related species and even within species? And the answer to this data traditionally was to focus on morphology and other sources of data, but especially morphology. But beginning in the 1970s and 1980s, we began to be able to use DNA sequencing to address these questions. And this really was a revolution in our understanding of evolutionary relationships. And so what you see here is, as far as I can tell, and I may be wrong about this, but as far as I can tell, the original or the oldest DNA-based plant phylogeny paper that was ever published in 1982 by Palmer and Zamir. And even though it's a relatively simple tree, it was such a big deal at the time that it was published in PNAS. And on the right is a map of a typical chloroplast genome from angiosperms. And this paper was actually, or this tree was actually generated by using restriction enzymes to chop up the chloroplast genome into bits and then run the bits on a gel and look at patterns on a gel. So this wasn't DNA sequencing per se, but it was using DNA. And as soon as we began using DNA, we began to be surprised by the results. And that is illustrated in this very first tree. This is a phylogeny of tomato, Lycopersicum esculentum. At the time, that was the name of it, and its closest relatives. One of the first things that we saw in the very first molecular phylogeny of plants is that traditional taxa were not monophyletic. So the S here is Solanum and the L is Lycopersicum, and you can see that neither one of these genera is monophyletic in this tree. So right off the bat, we're discovering that traditionally circumscribed taxa are not monophyletic. And not only that, in this tree, there's a species that's not monophyletic as well. Lycopersicum peruvianum occurs over here within a clade, but there are other species of Lycopersicum inside that clade. And, this is actually not unusual at all when you use molecular data. And that's one of the reasons I love this example so much is that the very first tree presaged sort of many of the issues that would become common later on as we continued to use DNA. Throughout the 1980s, more and more plant systematists began to incorporate DNA sequencing into their own work. And a lot of the early studies focused on deep level relationships, so sort of family level relationships, ordinal relationships, things like that. And they focused especially on a particular chloroplast gene, RBCL, such that 11 years after this Palmer and Zamir paper came out, there was another major milestone paper, Chase et al. 1993, which was the first phylogenetic analysis of basically all families of seed plants. And that was a really big deal. It was over 500 taxa. It required a small army of plant systematists to do this. And I think, that was a spirit of collaboration, really, that has become a hallmark of plant systematics. And it really was a remarkable achievement for the time. It's still a very highly cited paper to this day. So after this paper came out, there was a lot of optimism in plant systematics that we were going to be able to figure out plant phylogeny. And we were going to march to the tips, all the way to the species at the tip of the tree. And we were going to figure all this stuff out with, a few genes, potentially. But as we moved into the 2000s and we were DNA sequencing became more feasible. We could get larger data sets. Some pretty significant challenges became apparent, particularly among closely related species, like you would see here in the genus Schedia. And so what I want to do today is talk about some of these challenges and then talk about how genomic data might help us sort out these challenges. And then at the end, I want to talk about how all of this can be helpful to conservation, which is very, very important. So let's start off with just reviewing a couple of the big challenges with using DNA sequence data to understand evolutionary relationships. Okay, the first challenge is that there's gene flow everywhere. Now, we've always known that plants were good at hybridizing, but now we know that all organisms are good at hybridizing. So there's gene flow everywhere and every when, all through time, really. And so this is just, I'm going to show you a couple of examples of this from recent work. This is a paper that actually just came out a few weeks ago, and I love the title, Admixture May Be Extensive Among Hyperdominant Amazon Rainforest Tree Species. And I think this admixture may be extensive is probably true for almost all plant groups. So this is a group called Eschweileria, which is in Lecythidaceae. This is an American tropical wet forest group. And this is a structured plot. Maybe you're familiar with this, maybe you're not. But in a structured plot, each column is an individual. And we're able to estimate the proportion of ancestry from different clusters, however we set those clusters up, within an individual, and that's what the different colors are. So this is a cluster plot, a structure plot based on 344 nuclear genes. So it's a lot of data, and this is becoming increasingly feasible. And with this much data, you have pretty good confidence that individuals like these two that have stars on them are probably hybrids between Eschweileria coriacea and Eschweileria wachenheimii. That's why there's half purple and half green, and so forth. There are many individuals here that appear to be admixed. Here's another example of this. This is a slightly different way of looking at things, and this is from European white oaks, which actually have been worked on quite extensively. In fact, we have good genome sequences for these species. And because of that, we're able to zero in on specific SNPs. A SNP is a single nucleotide polymorphism. Basically, it's a variable base in the genome. And we know where they are in white oaks because we have really good genome data. And so we can literally just look at the 8,000 SNPs that are known to vary among these species and estimate all kinds of interesting things about them. But this here on the left is a tree. It's in 3 dimensions. The y-axis is time, and the other 2 axes is just so that we can visualize gene flow among the lineages a little better. But they have different time events, split 1, split 2, split 3. These are just phylogenetic splits in the tree. And what's really interesting is down here at the bottom, we have this red sort of slice through the graph that is TSC, time of secondary contact. So using this SNP data, these researchers were able to estimate that all 4 of these species have hybridized recently with each of the other species, which kind of makes sense for wind-pollinated common tree or a group of common tree species. But they were also found evidence of secondary contact in the past secondary contact. They weren't able to really determine exactly when that was, but there was evidence of earlier hybridization. And the point of all of this is that if you can get an accurate phylogeny, and if you have that genome sequence, it can provide a really large amount of insight into patterns of gene flow, past and present. So it's really very, very, very valuable information. Now, the second challenge is related to genetic drift. And I'm sure you're familiar with genetic drift in population genetics, but it also can create issues for phylogeny estimation during speciation events. And there's a phenomenon called incomplete lineage sorting, which we always abbreviate ILS, which happens during speciation events. And this figure is a really nice summary of what happens or what incomplete lineage sorting is. And basically, what you see here on the left is a species tree of 3 species. And what's nice about this is that it's showing each species as a population. So every ball in this diagram is an individual organism. And every row is a generation, right? And the different colors are different alleles. So we can trace the ancestry of every individual in this diagram. And really the alleles is what we're interested in. And so the problem with incomplete lineage sorting from an analytical standpoint is that it allows for sequences, identical sequences, to be shared between species without hybridization. So if you look at this green lineage here, right here, you can see that for a time, this green allele is present in 2 of the daughter lineages. And so during speciation normally happens by populations splitting apart. And populations are composed of variable individuals that might have, as a population, many different alleles at a single locus, right? And so some of those alleles at that locus may descend into one daughter lineage and some may descend into another. And some may descend into both. And when that happens, you can pick up the exact same sequence later on in time. You can pick up the same exact sequence in multiple lineages. And so if we were to take a time slice right here at this generation and reconstruct the phylogeny right here, this is the tree we would get. And we would look at that tree and we would say, wow, these species aren't monophyletic because I'm getting exactly the same sequence in different places in the tree for many of the alleles. But it's really important to remember that these species are actually, in fact, monophyletic. If you look at the species tree, you can see that, no, all of these individuals are from species B, and these are from species C. But when you reconstruct it here in this point in time, which is very, very close to the speciation event in time, you actually reconstruct a gene tree that shows the species are not monophyletic. And in fact, they are. If you actually waited longer in time, obviously we can't do this in practice, but given enough time, evolution will occur, mutations will happen, et cetera, and the species will then appear monophyletic within the gene trees. But that takes time. So if you're very close in time to this speciation event, you're more likely to see this, incomplete lineage sorting causing non-monophyly in a gene tree. And so gene trees and species trees are usually not identical. And just to emphasize, when we do molecular phylogenetics, we go out and sequence DNA. And then we build a tree from that DNA. And that gene thus has a tree. And that gene tree may not be the same. In fact, it's probably not exactly the same as the species tree is, whereas the species tree is the actual series of splitting events that happened in the populations. And we would need a time machine to be sure of that, right? To actually go back and see it happen. So in practice, what we're doing in systematics is taking a bunch of gene trees and trying to figure out what the species tree is. But the gene trees aren't usually the same as the species tree. And I want to give you an example of that from my own work. I do a lot of work in the desert Southwest, particularly the Chihuahuan Desert over here in New Mexico and Texas and Mexico. And I work on plants that grow on gypsum a lot, deeply in love with gypsum plants. And this is some work on the genus Annulocalis, which is illustrated here. This is a Nyctaginaceae genus. And these have huge flowers, beautiful plants. They're hawk moth pollinated. I could go on and on and on. But there is a clade within Annulocalis of 2 species that are only found on gypsum. And most of one species is very narrowly distributed, and the other one is very broadly distributed, and that's Annulocalis leosolenis. And so over here, you see a tree, and the colors in the tree, the colored boxes, correspond to the gypsum endemic species of Annulocalis. And the colors in this distribution map correspond to that phylogeny. And so you can see there are discrete groups, geographically speaking, of gypsophiles in this genus. And what's really interesting is that morphologically, all of these things are very similar. They're very, very similar. They're clearly very closely related to each other. But in this chloroplast tree, based on chloroplast DNA, the populations from central Arizona, which are in blue here, are not grouped with the rest of the gypsum endemic ones. Instead, they're grouped with another species of Annulocaulus, which lives only in Death Valley and the immediate area around Death Valley and is not associated with gypsum whatsoever. And so this makes no sense geographically or ecologically. It's very, very strange, but it's a very strong pattern. But if I look at a different gene tree, a tree based on nuclear data, well, lo and behold, all of the populations in this entire group, and their colors aren't quite the same, so I apologize for that, but all of the populations in this entire group form a nice, well-supported clade, including the central Arizona populations. So 2 gene trees give you 2 very different answers. How could they be so different? They're from the same individuals. How could they be so different? Processes like gene flow and ILS. Which brings up a question, how do we discriminate between them in practice? How can we figure out whether we have one or the other or both? And that's actually kind of a difficult question. And I will just say that to really get at the answer to that question, in brief, 1 is not enough. And what I mean by that is 1 population per species is not enough to address this question. So imagine if I had only included one individual of Annulocaulus leosolennis and 1 individual of the other gypsophile Annulocaulus reflexus. If I had picked this leosolennis or that leosolennis, and that was all that I had picked, I'd get 2 very different trees. So 1 population is not sufficient, and 1 locus is not sufficient. 1 gene is not sufficient to resolve all the complexities of evolutionary history. And so really sequencing lots of loci is necessary if you really want to drill down and understand what's going on in your favorite lineage. So I want to circle back to the beginning of the talk when I showed you a tree about tomatoes. This is a tree from 2016 based on 2,800 gene trees, nuclear gene trees. And so here is tomato there. This is a little bit larger of an analysis in terms of taxon sampling, but it's a lot bigger in terms of locus sampling. And in an economically important group like tomato, we have the benefit of having very good genomic resources. And so we can do really, really cool things like sequence all of these genes for all of these species and then map where the gene tree is on the genome, which is, I don't have more time to talk about that, but that's really cool stuff. And just before I go any further, let me say that this is a lot of data. And I won't say that it's easy to generate this data, but it's certainly a whole lot easier than it used to be. Like we wouldn't have dreamed of doing this 20 years ago. And now it's becoming more and more routine. And so the talk that follows this by Norm Wickett and Angela McDonald will go into more details about how we get these kinds of data. But to circle back, when we have this much data, we're actually able to start to discriminate between gene flow and probable incomplete lineage sorting. So I just want to give you a brief example of that here. So this is a really weird looking tree over here, and this is a more normal looking tree. So what this is the tree that is generated if you took all of these data, glommed them all together, and treated it as a single gene. This is the tree you would get. And the authors were using this as an approximation of the actual species tree, which it may or may not be. The only way to be sure of the species tree would be to have a time machine, which we don't have. Whereas on the right, this is actually all of the gene trees superimposed upon each other. And it's created by a program called Densitry, which I think is a great name for a program. But it allows you to sort of do a visual analysis of patterns. And so you can see, in some cases, there's just a massive, a dense tangle of everything. You can't really detect any consistent gene patterns. But here you can see all of the gene trees seem to support the existence of this clade. But once you get inside that clade, gosh, you can't really detect anything. Over here's another really interesting thing. Here's a species called Solanum wylacensi. And this particular individual had a lot of gene trees that attached to this individual as Solanum peruvianum, and then another set of gene trees, a lot of them, that actually showed this individual as sister to this whole clade. So there were two signals, basically, among different gene trees. That's interesting. But these patterns make sense in light of breeding system. So all of these solanums, including tomatoes over here, are selfers, whereas this clade, these are obligate outcrossers. So we might expect to see a lot more gene flow involving these species compared to that. And if you look carefully at this, you'll see lots of examples of individuals here that have sort of two dominant signals, which would be what we might expect with gene flow or hybridization. So really, really powerful what we can do if we have a lot of data, but we need non-molecular data as well. They matter a whole lot. We can't just have genomic data. We also need morphology, demography, which I think is often overlooked, and we need a lot more demography data. Pollination biology, physiology, ecology, chemistry, you name it. One of the wonderful things about working on Shedia is that Anne and Steve have spent 30 plus years generating an enormous data set of stuff like breeding system, chemistry, stuff like that, which allows us to interpret our molecular phylogenies in a much more powerful manner. And integrating all of these data together really helps us understand evolution, but also make better informed conservation decisions. And so I think knowing all of this, having this genomics perspective, along with all of the other sources of data, can really help us get through some thorny conservation questions. And I'll just point out a couple of them that I think genomic data might be really valuable for today. And one of these is something that I think is encountered pretty frequently in the Hawaiian conservation, plant conservation for sure, is should we actually take two populations within the same taxon that might be distinct in some way? So they might grow on different islands or even different parts of the same island. They might have slightly different morphology. Two different populations may have slightly different morphology or slightly different ecology. Should we actually ever hybridize these things if individual populations decline to the point where inbreeding becomes a problem, right? And that's exactly what happened in Shedia kauaiensis, which you see here. These are flats of seedlings from the National Tropical Botanic Garden, which is a partner with us in our Shedia project. There are very, very few of these left in the wild, Shedia kauaiensis, and individual populations have become inbred to the point where their fitness has been impacted negatively. Steve and Anne at Irvine have taken different populations, crossbred them, and the F1 individuals are much more vigorous growers and have much better seed set. And these are actually being reintroduced in exclosures on Kauai. So knowing the very fine population genomics, essentially, of species can be very, very useful in guiding these decisions. Another question that I think is something that we confront with and will confront more frequently in the future is what happens when we have populations of rare species that have hybridized on their own because we have allowed them to come together due to human alteration of ecosystems. So here's an example of this that is recent that I use in my first year seminar on conservation biology, which is about red wolves in the southeastern United States. There are very, very few of them left. They had already started hybridizing with coyotes when the captive breeding program was started in the 1980s. And then about 5 years ago, a team of biologists that was noticing a group of canids on Galveston Island, which is really not out of the way in any sense, it's near a major city, noticed a group of canids that kind of looked like red wolves and did some population genomics and found that they had a significant portion of the red wolf genome, and they had alleles that weren't found in the captive breeding program. And so there's some thorny sort of philosophical questions about whether we should breed things like this, but at least we know about them now. And that's, I think that's the point, is that conservation genomics is a tool, right? It's another tool in our toolkit. And I hope that I have been able to convince you that it's actually really useful, and that it will help us, for example, save all these endangered and threatened species that you see in front of you. This is actually the outdoor conservation greenhouse facility at National Tropical Botanic Garden, which I was very lucky to visit a couple of years ago and was very, very inspired by. So I hope that genomic tools can be useful in conservation in a real way. So that's it. I just stopped by acknowledging several people. I want to acknowledge NSF for making this possible, and I want to acknowledge Lauren Weisenberger and all the organizers of the meeting, Gary Krupnik, Chris Iskogan, et cetera. Thank you so much for putting this together and bringing us together today. I'd like to thank the rest of Team Shedia, which is who you see in front of you, for all your support and camaraderie. And I want to thank the Smithsonian and National Tropical Botanic Garden, and Nina Ronstedt for being a wonderful partner. Thanks to Matt Kira and DOFA and all the other conservation organizations, U.S. Fish and Wildlife, et cetera, that are all in Hawaii doing wonderful work. And really, I want to thank all of you who work so tirelessly to preserve the Hawaiian biota. Thank you very much.